So we are live now. Good evening, everyone. So I am Inder. Welcome to the third season of our ongoing third session of our ongoing third season of Weekend Chat with Researchers. We are Bugbears, a research group at NCBS headed by Dr. Aswin. Here, this is a yeah. So this is our science and research outlet channel, and we are back again this weekend with a new topic, with a new research, but with the same old dose of fun science. This week, your host Vidya and myself Inder, we are gonna start this session. Okay? So let's start with our wonderful poster designed by the Vidya.
Hello, everybody. I'm sure you've all had a look at the poster. It shows the mouse embryo as the blueprint for the complex body plan of its adult counterpart. So what do we know about how an organism develops? We know that every multicellular organism starts as a single-celled embryo. That the start of such a complex adult is a single cell sparks off many questions. How does the single cell morph into an adult? How is all the information about such a transformation encoded? Are there any similarities in this transformation process across species? So to answer all these questions and more, our guest today is Dr. Ramkumar Sambasivan. Ram is an associate professor at IISER Tirupati. His lab works on the mechanisms regulating the development of vertebrate embryos by studying signaling cues, transcription factor networks, and microRNAs in the commitment of cell fate and position-specific identity along the body axis. Welcome, Ram.
Thank you, Inder, and thank you, Vidya. Yeah, and thanks everyone at the Bugbears for putting this together. So should I start? All right, so let me share the screen. Okay. Is this fine? Can you see it? All right. Okay, cool. So once again, thank you very much for having me here today, and to share the sort of excitement that motivates the work in my lab, and this is going to be sort of, I'm going to keep it broad, yeah, and so let's get started. So as Vidya was saying, one of the thing that this field, developmental biology, addresses is what is shown here in this slide. So I would start off with this little writing that from 12th century that tells us what people were thinking in those days about how an animal forms. Yeah. So these were primarily monks, Christian monks, who were also interested broadly in understanding how the world works. And so the writing tells us that the thinking was that God sent tiny angels into the womb of every pregnant woman in order to sculpt the embryo. Yeah. Otherwise, this conveys this sense of wonder that one had, how this this animal is created. Yeah. So this is what this the developmental biology is all about. How the single cell, which is the egg fertilized by sperm, develops into an animal. Okay. So developmental biology, as we know today, deals with this part, which is the embryonic development, okay, that gives rise to this newborn that you see here. It also deals with the development that continues to form the adult, the sexually mature adult. And you also developmental biologists also study aging. So development is something that doesn't stop. It starts with the fertilization and all the way to the death. The aging process is also part of the development. And we study regeneration and disease as well. Okay, this is what developmental biologists broadly study. So what kind of questions the developmental biologists ask? Yeah, we'll focus on two very fundamental questions. One is the mechanism of cell differentiation, which is you have only one cell, but in the adult human body, there are up to 300 different cell types. Yeah, you have skin cell, you have hair cell, you have lens cells in the eye, muscle, blood cells, heart muscle, and so on. Yeah, you need to generate this variety of cell types starting from a single cell type. How do you generate this diversity? And also the question of morphogenesis. Yeah, how do you generate this shape? So this is what the mechanism of cell differentiation is shown in this schematic. Starting from this single cell zygote, you have to generate different cell types. Yeah, a few different cell types are shown in here. Okay. This is the question of morphogenesis that I was talking to you about, shaping the animal body. Okay, I'll tell you one sort of theory, talk about one theory which helps people abstract and understand what happens with shaping the animal body. How do you lay down the animal body plan or the blueprint, as Vidya mentioned? Okay, so this is a theory where this works like a Cartesian coordinate system. So this was, of course, comes from René Descartes, the French mathematician. So it is called Cartesian because it comes from his name, Descartes. And that, you can specify a given location in this field, yeah, based on the two axes. The coordinates can be specified based on this x and y axis. This is the typical Cartesian coordinate system. Now, you can imagine the early embryo as an embryonic field similar to this one, where this field is filled with cells, and these cells all have equal potential. Okay, and the location they occupy in this field will give them their identity. So if you're in this location, you're going to become heart muscle. If you're in this location, you're going to make kidney. If you're in this location, you're going to make brain, and so on. So in this coordinate system, it is x1, x and y axis, but in the embryonic field, the axis becomes the head-tail axis, also referred to as anterior-posterior axis, and also the back-belly axis. Okay, so like these numbers that you use to provide the coordinates, the cells need to compute the location that they occupy. Okay, and this location, information of location, must be given to those cells so that they can acquire a particular identity. And that information is present in the form of signals present along these axes, the head-tail axis and the back-belly axis. Yeah, typically, some of the idea that you may have is that you can imagine a particular molecule that is being generated here, and it is secreted from the cells from one end, and simply by diffusion, it establishes a concentration gradient. Okay, and so you have two such concentration gradients along these axes, and you can compute the location based on these two information, very similar to this coordinate system.
Ram, I have a question regarding this.
Yeah.
Yeah, so in maths we know like where to take the origin. Like we know that 0, 0 is the origin. But if embryo is just like in embryo we have this collection of cell after association. So how do we say that this position is the 0, 0 for that? This is the origin.
Yeah, so very good question. I'm not sure we have something similar to origin in this case. This is also the Cartesian coordinate system is a little bit of oversimplification, I should say, of what we know. In addition to this, for example, concentration gradient, let's say a molecule is being secreted from here. It will establish a diffusion, but simply by gradient diffusion, it will form a concentration gradient. You can also have another molecule that is antagonistic to this, that is forming a counter gradient, right? So there is a lot of complexity in the system, but to specifically address this question of origin, I don't think there is something similar to that can be that we understand in this embryonic field. Yeah.
Ram, I also have a question.
Sure. Yeah.
So before the establishment of these chemical gradient axes, the embryo is equivalent from all directions, right?
This is another important question. So we do know for a fact, based on experimental evidence, that up to some point that the embryo is equivalent, every cell in an embryo is equivalent. It is totipotent. It has the capacity to make any cell type. But you also would have heard pluripotent, right? So even at a later stage, when in that stage, for example, the human embryo looks like a disc. Yeah, and in that disc, the pluripotent cells, before the beginning of this process called the gastrulation, they are all thought to have equal potential to form any cell type. Okay, but once gastrulation starts, which is when the axis information is being established, then you immediately have different fates that are being formed. Okay, so yes, the answer to that question is yes. At just before the formation of axis, it is a state called a pluripotency state, and so at that state, all the cells have equal potential. Yeah. Okay. So let me continue here. So why do we study developmental biology? I thought I will address this right at the beginning. Okay. How does it help? Of course, a scientific enterprise is the motivation is knowing the world around you. It is curiosity driven, but in addition to that, practically speaking as well, this knowledge of developmental biology is key to design stem cell differentiation methods. Okay, so this is actually, as I would tell you at a later stage, is driving huge investments into developmental biology today, right? And a lot of interest in studying early development. That is because if you want to generate a cell type of interest starting from stem cells, yeah, these are the pluripotent stem cells, the induced pluripotent stem cells that many of you may know about. Basically, these are cells derived from patients themselves, for example, and you rewind this. So development takes the undifferentiated cells, embryonic cells, towards particular cell type, right? Through a series of gene expression changes, right? Gene expression states, right? So you can rewind this back by using some tricks in the lab. You can rewind it back and take it to a pluripotent state. The state that I told you is the equal potential to form any cell type. Okay, and these are known as induced pluripotent stem cells. Okay, so if you want to take these patient-derived induced pluripotent stem cells and differentiate them towards any cell type of interest, like for example muscle or blood cells, this can be used to treat diseases. Yeah, this can also be used to study the disease itself, mechanism of disease itself. Yeah, for this you need to know developmental biology. But how will you guide these cells to form a cell type of interest? You need to know how it is done in the embryo, how the embryo does it, and you try to mimic that in the dish in order to get the cell type of interest. So this is where there is renewed interest in early developmental biology, and people are focusing on this. Yeah.
I have a question.
Sure.
So the rewinding that you mentioned, the redifferentiation, is that something similar that happens in cancer cells?
That's a very good question. Yes. The rewinding does not happen all the way. Yeah. Only in case of teratomas that happens. That is the yeah. But most cancers, the rewinding is happening partially. Yeah. Yeah. That's a very good question. Yes.
I have one more question, but it's more on application side.
Yeah.
The embryo development is in a three-dimensional space inside the womb and all. And when we are developing, like, or we are culturing the cells, any type of, we are doing it on a petri dish or in a flask kind of situation.
Right.
So how do we dictate to take it a shape or anything like that?
That's a good question. Actually, this is what it brings me to the next point. Actually, how do you make organs? So, in terms of therapy that you think of, one can also think of organ replacement therapies, right? For a kidney, for example, or islet cells in case of, diabetes and so on. Okay, in that case, you need to have a three-dimensional organ, not a two-dimensional cell layer, right? So this is also that people have started now doing. Okay, this is not advanced to a stage where we can generate organs in the dish, but this represents a breakthrough in this direction, which is the organoids. Okay, you can push stem cells to organ-like rudiments. Okay, and that is sort of revolutionizing disease research and medicine. So the organoids that are generated from stem cells, they're rudimentary organs that you derive from these induced pluripotent stem cells. Okay, and they basically 3D organ-like tissues, and they have different cell types of the organ. And the way you generate is you do not allow them to attach. You keep them suspended in the medium. Yeah. In some cases, you also provide this gel-like medium in which you suspend the cells. This is jelly-like. This jelly is made up of proteins that form the extracellular matrix normally in the tissue. Right? And when you embed these cells in these jelly-like tissues, or you simply suspend them in a liquid cell culture medium, they actually organize themselves to take these three-dimensional space. Importantly, they form different cell types present in an organ, and the spatial relationship in terms of spatial arrangement, the relationship is also retained as in an organ. So you recapitulate the spatial organization of an organ in the dish. Yeah, it is because all that information is present within the cells, right? So they self-organize, and so it is possible to do that as well. Now the challenge is actually to take these rudimentary organs and really develop the life-size organs that can be used for therapy. Now that's where the field is going. Okay. So
Ram, we have a question from Harin.
sure.
So her question is, in cancer, what makes the cells go bad? In other words, what makes the cells do things against your body, where in the normal embryonic development, they're actually behaving the way they're supposed to?
Mm-hmm. Yeah. So they are uncontrolled in a sense. They are proliferating and growing where they're not supposed to, right? And so if you take, for example, in the brain, growing a tissue continuing to grow in the wrong place could disrupt the tissues around the neurons around. The neural circuits can be disrupted because it'll go and press against, right? So yeah, so growing in the wrong place and uncontrolled growth is the primary problem per se. It is and the problem is also metastasis. Even if you have a malignant tumor, in one place which doesn't bother you really, it can metastasize and grow in a place that it could become problematic, right? So basically cutting off tissues, supply of blood, or pressing against a nerve, or lots of things can go bad when you have this. So the cells, yeah, this is what I understand of what happens. Yeah.
So, so then the chemical gradient which determines the axis and cell fate superpositions, which is present in the embryo, is not present in the adult. So there is no limit.
So, yeah. So as I said, these are again partial rewinding. It does not go all the way, and yeah. So this is, so in some sense there is recapitulation of what happens in development. So cancer is also considered to be, development gone wrong. You are basically reenacting things that happen during development at the wrong time, in the wrong place. Yeah. But you don't have the entire pathway that is happening. Even in some cancer cells, the question is, what cell gives rise to that cancer? The cell of origin, that is the question they ask. And depending on what type it is, if it's a stem cell that is giving rise to cancer, you will also see some differentiation happening. So it will enact the developmental program to some extent. But its program will be disrupted because of the cancer causing mutations. And so it will be different from normal developmental scenario. Yeah. All right. So now this is what I told you about. So the organoids are actually revolutionizing disease research and medicine. Just a couple of examples to make this point. So when Zika virus was spreading through, and people found that the babies born to mothers infected with Zika virus displayed microcephaly. The brain size was small. Yeah, they have very tiny heads, and the brain size is really small. Now, it is difficult, especially when it comes to new viruses, to understand whether it is really the cause for microcephaly, and if it is the cause, how does it do it, right? it is difficult because the virus will not be infecting any other species. So you may not be readily having an animal model, right? So how do you go about addressing this question? In this case, the forebrain organoids that people generated in the dish, starting from stem cells, you generate organoids that looks and has many cell types of the forebrain region, and then infected them with the Zika virus. And they showed that Zika virus specifically goes and targets some of the neuronal progenitors, yeah, and cause them to stop proliferating and differentiate faster. And when you do that, of course, if you deplete the progenitors early on, the size will be reduced, right? Because the progenitors will have to amplify to increase the cell number, and so you will reach the correct size of the organ. But if these progenitors are depleted early on because of this viral infection, then you're going to have a smaller brain. So this is something that was beautifully shown, a few years back using forebrain organoids. Without this, it would have been very difficult to establish that the Zika virus causes microcephaly and how does it do it. And of course, this is something that is making a big difference to patients with cystic fibrosis. There are a number of drug molecules available, and cystic fibrosis is also caused by variety of mutations in that single gene. So depending on where that mutation is in that gene, one or another drug will be useful. So when a patient walks to the clinician with a new mutation, what drug should be used for that patient is difficult to tell. And you cannot try different drugs until the patient responds and the disease is ameliorated. So now people are beginning to use organoids from the rectum of these patients, and they have developed an assay to screen the drug that will work. And this is now being used, at least for one patient. It was very successfully used in Denmark, and they're trying to expand this and offer to patients in Europe now. Okay, and the most striking is this eye-shaped cup. This is how our eyes will look like. This is the retinal tissues that you see. Okay, the pluripotent stem cells can be guided to make these retinal or optic cup organoids in the dish. And in Japan, they are going ahead using this to treat macular degeneration in elderly age-related macular degeneration. So yeah, so this is not just fiction. So how do you develop all these forebrain organoids or optic cup organoids or rectal organoids? You need to know developmental biology, how to guide the pluripotent stem cells to get these particular organoids of interest. Yeah. So this is why developmental biology has gotten a lot of attention these days.
So Ram, I have a question. So when we are taking the stem cells or the induced pluripotent cells, so and we are making them into organoids or we are differentiating them, is it I think it's kinda like a controlled cloning, because if we are making a forebrain of someone, then that brain would be of someone which we have taken
Right.
samples from.
Yeah. Yeah. It is in a sense, but you're just making only part of that person, right? So ethically, there are no concerns here. And in fact, it is advantageous because when you go for allotransplantation, which is what is done, right? The graft rejection is a problem. If you take the cells from the patient, yeah, correct the disease-causing mutation, and then make the cell type that needs to be put back in, or the organ that needs to be put back in using the patient-derived cells itself, then the graft rejection problem is alleviated.
Oh, because it's just like a clone thing.
It's a clone, yeah, but it's only part of the, yeah. That's a good point. Okay. So so I told you so far, what is it that we study in developmental biology, and also why we study it. What is the sort of practical, application that comes out of it, in addition to satisfying one's curiosity. Now, I'll tell you a little bit about how do you study embryonic development? Yeah, what kind of approaches people use? What do developmental biologists do in their lab? Okay, I'm talking about majority of the developmental biologists, but there are developmental biologists also come in different flavors. But we will stick to the most the vanilla flavor developmental biologist. So, if you start off from the historic perspective, early developmental biologists were all embryologists. They took embryos and they compared embryos of different species, and then they followed which part of the embryo is giving rise to what tissue. And so this was primarily embryology. They did comparative embryology. And in fact, 19th century, the comparative embryology that was done by a few people provided key evidences for evolution that Darwin used later on, to formulate his evolution theory on evolution. And in early 20th century, people started little more sophisticated experiments, taking the embryo, removing one cell by just killing it, yeah, or trying to dissociate one cell to ask what that cell gives rise to, and so on. But early 20th century also saw the development of field of genetics. And this actually, the genetics was born from the field of developmental biology, because early geneticists were all developmental biologists. Then came the understanding that that development is driven by genes, right? And there are key developmental genes which regulate the process of development, and this marked the birth of developmental genetics in the mid 20th century. And from then on, the major way in which developmental biologists have been doing studying developmental biology is through developmental genetics. Yeah, still there is embryology, still there is comparative embryology. All those flavors exist and they contribute significantly to the field. But this is the major way in which people are doing, right? So I'm just giving you some example. Yeah, so Hox genes are the genes that I told you about, giving information of position along these body axis. If you take the head-tail axis, the initial information in terms of signaling cues is translated into expression of specific genes. Yeah, one set of genes that is very important in giving positional information along the head-tail axis are known as Hox genes. Okay, so so when you this is how the normal skeleton of a mouse looks like. This is a skeletal preparation, and rib cage is the ribs are formed in the thoracic segments, vertebral columns, right? So up to 13 vertebral segments, you will see formation of these rib bones. But in the lumbar segments, yeah, after the 13th segment, you do not form any ribs. And the sacral segments, which is after the lumbar segment comes, you will find structures that you do not find in the lumbar segment. So these segments actually mark distinct positions along the head-tail in your vertebral column, head-tail axis in the vertebral column. Okay, so if you mutate HOX10 genes, yeah, if you do loss of function of these, which is basically the developmental genetics experiments, you remove these genes from the embryo by genetic experiments, you see what happens. Beyond this T13, what should have become lumbar and sacral vertebral segments also begin to develop rib bones. Yeah, So basically, the identity of these vertebral segments are now transformed to that of more anterior segments, that is rib forming thoracic segments. Okay. And if you remove HOX11 genes, here you removed HOX10 genes. If you removed HOX11 genes, the sacral segments begin to look like lumbar segments. Right? So this is one type of experiment where you remove the genes and try to see what it does to the embryonic development. Yeah, and therefore try and understand what kind of genes control given developmental program. And here you're looking at misexpression. This is expressing a gene where it is not expressed normally, right? So this is, for example, if you misexpress HOX6 gene, one type of HOX6 gene, throughout the vertebral column, you form ribs all along. Yeah. And if you misexpress HOX10 that was removed here, all along the vertebral column, everything is lumbar segment. Yeah, you don't have any thoracic segment at all, so no ribs are formed. Okay, so I'm kind of doing two things from this slide. One is to tell you how genes are contributing to giving information of position along the head-tail axis. Yeah, the other is to also tell you how these developmental biologists actually go about investigating the process of development.
I have a question.
Yeah.
So you said that HOX is one of the chemicals which establishes the gradient. Is there a cause for this redundancy? Like ideally, like hypothetically, two chemical gradients should establish the whole Cartesian plane.
Right. Yeah. So there are primarily, if you look at from the evolutionary angle, wind gradient establishes the head-tail axis, and BMP gradient establishes the back belly axis. This is fundamental, and it is conserved across all animals that have bilateral body symmetry. Okay, a majority, there are of course exceptions in biology. Okay, HOX is something that is switched on in response to this wind gradient. Yeah, so HOX10, as you can imagine, will be expressed only in this part. From this part onwards, lumbar segment onwards. So once HOX10 is expressed, the animal knows this is where I should stop making ribs. Yeah, because we are coming into another territory, so lumbar formation begins. HOX11 expressed, it knows now it is the sacral segments to be formed. That's why when you remove HOX11, everything looks lumbar. When you remove HOX10, everything becomes a rib-like. Yeah. So this boundary establishment fails when you remove the HOX genes. These are transcription factors that are switched on in response to wind gradient. Yeah. But there is there is a lot of complexity. There is a lot of redundancy in who regulates HOX. Yeah. That's because, you know, biology is always heading towards complexity, right? So
there is a question by Harini. Is it dangerous for the mouse to have ribs in all places?
So these animals will not survive. So they go on and they form these you know, ribs all along. So until birth, you can come. After birth, they will most of them will die. So these will be lethal, because what is shown here is how the one particular structure is affected. Yeah, these animals may have other defects in other organs, and so they are usually lethal.
So there's a follow-up question to that. So then the question is by Harini, that are these experiments possible for human embryos?
So yeah, these are no, the even to do this in mouse, we need to go through ethical clearance and the ethical oversight. In humans, there is no way we can do these. We cannot do experiments on humans. Yeah. So these can be done in mouse. It's also easier to generate a lot of these animals. Yeah.
I have a question. You said that usually the mouse dies when we do this kind of studies and mutation. Has there any case where the it was advantageous for the mouse embryo? Like if any like has ever occurred?
So these are dramatic changes that we bring on. Yeah. So, but there are always, of course, advantages. So if you look at the loss of limbs in the snakes, that is again due to changes in the oxygen expression pattern. So evolution has also made use of this. And the number of neck vertebrae is different between birds and mammals. Yeah. So these changes also happen because of changes in the oxygen expression. So of course you can play around with the oxygen. The evolution does that to create variations in the body plans and different forms. Yeah, that we see on Earth. But the kind of manipulations that are done here are way too severe. Yeah. Pretty dramatic changes. Yeah. In fact, it is major changes in hawks can be pretty detrimental as well, because in people, we have 7 neck vertebral segments. Any change in this number is usually associated with severe forms of cancer early on. So you always see this strong correlation between people with in more number of neck vertebral segments and some type of cancer. So these changes are very rare. So some changes that are accepted, and so you have changes that bring about evolutionary changes, but this is very fundamental to body plan. Okay. So by also continuing this idea of looking at ways in which people study developmental biology, there has been some another sort of revolution in the field of developmental biology that's happening in the last few years. We are witnessing it, and that I will come to. Yeah, what one can call as a renaissance of developmental biology. As I mentioned early on, to exploit stem cells in disease research and medicine, in order to drive the differentiation of pluripotent stem cells into cell type of interest, or to make organoids, we need to have knowledge of embryonic development. So this is the driving force. And what people have done in this in the last few years, that is this renewed interest has brought in a lot of technological advances. One technological advance that is being now applied in the field of developmental biology is studying development at single cell resolution, right? So what people have started doing is to get single cells, take embryos, dissociate to get single cells, and sequence the RNA expressed in these individual cells to understand the gene expression state of individual cells. Yeah, and now so this basically gives you an unprecedented resolution of differentiation mechanism. Because cell differentiation, you can see it as a sequential change in gene expression state. Yeah, so eventually you give rise to one particular differentiated cell where specific set of genes is expressed, and therefore it performs a specific function, right? And so now you have a great resolution into this differentiation mechanism in the embryo. Yeah, because of this technology. And it also gives you a holistic view of differentiation trajectories that is shown here. So you can take embryos, this is zebrafish embryos of different stages, isolate single cells, you just sequence them, and you just identify what cell types they are based on the gene expression pattern. You can see how gradually this early embryonic cells sort of diversify and form different cell types. Okay, so this is something that is making a big splash in the field of developmental biology. Okay, currently.
I have a question again. I just wanted to ask, like, uh, for when the cell is differentiated, it has the information to go back. That's how we are able to make it into induced and all.
Mhm.
But why does the cell loses the ability on its own to differentiate into its own, or like going? Because thinking from a like a animal perspective, isn't would this be much more advantageous? Like even if your finger get cut off, the rest of the cells just grow up to the stem and all. Mm.
This is a very good question. So this is what we call as de-differentiation. So we are rewinding it by doing some tricks, which is adding specific transcription factors and pushing them back. But in some animals like salamanders, this process naturally happens when they lose a limb. They're able to regenerate their entire limb. Like the lizards, when they lose the tail, they grow the tail. Salamanders have phenomenal regenerative capacity. They can regenerate their limb as well. And in some cases, this capacity comes because of their ability to de-differentiate. So differentiated cells go back, become stem cell-like, and then contribute to the growth of the arm that has been lost, the limb that has been lost. Okay, but this has been lost in human beings, of course. Yeah, why don't mammals have this capacity that amphibians have? That's a very good question. We don't have the answer to it. Of course, there are theories. The theory is that this is a trade-off. We do not want cancers. Yeah, the danger of developing cancer is high because that's a partial rewinding, right? So basically, we have lost our ability to do this great regenerative potential we have lost, by trading it off with protecting ourselves from getting cancer. Yeah, this is the theory in the field. All right.
Then this kind of implies that salamanders have a higher rate of cancer.
That's a good point. And yeah, one is people are actually trying to understand what other mechanisms it has to protect itself from developing cancer, right? These are all, the evolutionary trajectories, and what you see is at the end of these trajectories, right? And so going back and looking at them may help us also understand how they are balancing these two very well. Yeah.
Ram, we have a question from Harini.
Yeah.
So the early developmental trajectories in zebrafish embryos that you talked about, have people tried the same experiment in other animals? If so, were the results same or different?
Yeah. So that's a good question. So people have done it with fly embryos. People have done it with mouse embryos. A data set that we use, that's out there in the field and that we use. People are even beginning to do it with human embryos, because whenever there is aborted embryo, and people are willing to donate it for research, and that is being also used to generate this. And what people are doing now is to compare across these species, yeah, to understand what is common and what is different. Yeah, this in fact actually brings me to this. So this is one of the points that I was going to talk to you about, this cross species comparison of single cell data is causing another sort of rebirth of another field called EVO-DEVO, or evolutionary developmental biology. Okay, so I'll come to that. So, but this is just to sort of tell you more about the impact of the single cell technology on developmental biology, is that, eventually we need to understand what happens in the single cell approach. You take the embryo, you dissociate. So you lose all the spatial information, but you get what individual cell is all about in terms of its gene expression state, what types of genes it expresses, what it does not express, and so on. But you need to map it back to the embryo, right? So people are now using this information that they get from the single cell sequencing, doing RNA-scope, which is to look at gene expression in the embryo, to map it back to find where this particular cell is located in the embryo. But now we need to do this in a big way to understand every single cell in the embryo. This is mid-gestation embryo, mouse embryo that you're looking at. Okay, these are the forelimb. This is the hind limb that is developing. These are the segments that contribute to the segmental body plan, forms the vertebral segments, for example, right? So imagine in a few years, if you can pick one cell from here, one cell from here, and know what is its gene expression state, what types of genes it expressed at that point in time, in that spatial location. This would give us a complete picture of development. What kind of genes it is expressing, what kind of signals it is secreting to influence the neighboring cell, to what cues it is responding to from the neighboring cell, what it is going to do in the next step, in the 2 hours later, what it will become. Yeah, this kind of understanding is what developmental biologists hope to have with these exercises, that with this type of approaches that's happening currently. So as you can see, this is a very exciting time to be a developmental biologist. So yeah, that's the idea of a virtual embryo, map individual cells from single cell data, recreate the spatial information, so you know every cell in the embryo. Yeah, so that's where we are heading to, and this is what I told you about, this comparing across species. This single cell data is also sort of helping us understand evolution. Yeah, so this is a field called evolutionary developmental biology, colloquially EVO-DEVO. So actually, this is the point I will also tell you why understanding development and evolution are related, right? So your single cell data from embryos of different species, by looking at it, you get idea of evolution. But how do we say that? What is the connection between development and evolution? Okay, so in a nutshell, evolution is survival of the fittest. Yeah, development is basically arrival of the fittest. EVO-DEVO, this is what you talk about. So even though genetic mutations underlie evolution that lead to evolution by natural selection, development is the agency that brings about these developmental changes. Yeah, so I'm exemplifying this with this help. This question: how did the bat get its swing? Yeah, we have this arm. Yeah, it's a human arm. You can see it's the same set of bones that a bat wing also has. Yeah, so it's basically a structure that is homologous to the human arm, but it does not look anything like a human arm. It's a wing. It serves a very different purpose, right? So what led to this? How did the bat get its wing? Okay, so this is how the bat limb and a mouse limb looks like to start with. Early on, this is a developing limb bud. Okay, this is what is going to give rise to the wing. You can see these are already the digits that are going to form. These are the digits that will form. And between the digits, you have this web-like tissue in the early limb bud. At later stages, you can see gradually this web-like tissue is disappearing. But this web-like tissue is retained in case of bats. And you can see the different growth trajectory now. Yeah, the digits are elongating. The web-like tissue is maintained and it's also growing further. Okay, so basically there are expression of two signaling components, FGF and BMP4, is regulated by mutations either in their enhancers or upstream. Okay, so the change in the expression of these two genes and or their genes that inhibit their function and so on will result in retention of this interdigital web and continued growth of this to generate these type of long digits and the wings. Okay, so development is the agency which converts these mutations into morphological changes that then has an adaptive significance for the organism, and therefore that can be naturally selected. Okay, gives rise to a phenotype that can be naturally selected. Okay, so that's the sort of connection between development and evolution. Okay, so I thought I will tell you a little bit about vertebrate, my interest, one of the interest in my lab. So before we sort of close out, how much time do we have?
You can continue. We have on good time.
Okay, all right, cool. So our broad interest is to understand, this how positional information is generated along the anterior posterior axis. Yeah, specifically in one of the germ layers, group of embryonic cells that gives rise to skeletal muscles in the body. Skeletal muscles, heart, and some connective tissues and so on. Okay, so this is the major interest. But looking at this, we are also getting sort of into this is a new interest in the lab, which is sort of which I find very exciting, that we are picking up this problem to work on, is the evolution of vertebrate neck. Okay, I'm not an evolutionary biologist, I'm a developmental biologist, but by trying to understand neck development, we will be able to address the question of neck evolution, okay, through this evo-devo type of approach. Okay, so neck is something interesting. So initially, if you look at our vertebrate ancestors, yeah, they had their skull directly attached to the shoulder girdle. Yeah, when you had the shoulder girdle uncoupled from the skull, uncoupled from the shoulder girdle, right, you got yourself a neck. This gives you the possibility to move the head independent of your body. This became a huge advantage, especially with the animals moving into land, the terrestrialization, right? So moving around the neck, could mean life, difference between life and death. Yeah, you can see or hear your predator being close by, or sense your prey or whatever, right? So that's the sort of adaptive significance of neck. And so it is thought to have happened, this separation, this formation of this bone, excuse me, separating skull from the shoulder girdle, seem to have happened at the base of the vertebrate lineage. So as the vertebrates emerged at the very base of this tree of vertebrate, this seemed to have happened early in the vertebrate lineage. And so how did this new tissue develop? So not only do you have to form the bone, you also have to develop now muscles that will allow the movement of the head independent of the trunk, right? So our interest is, where do these new neck muscles come from? These muscles weren't there before, but with the emergence of this neck, you got yourselves new musculature that just permitted you to move the head independent of the trunk. So something new comes from in the evolution, a set of tissue emerges during evolution. The question is always, where does it come from? Nothing comes from nothing. It has to originate from something else which existed before. It's a modification. It's an elaboration of something that existed before. So this is what we try to understand. But understanding that question, where it came from, we will be able to do so by addressing the developmental biology of the neck muscles, which is what we are doing. So summarized here is some of the work that we have done also, and other people have done in the field. So these repetitive units that you see, yeah, this is actually muscle that is being formed in the mouse embryo. This is slightly past mid-gestation, 2 days past mid-gestation. Yeah, this is how musculature is being formed, and these are formed in this repetitive units, okay, that we call as somites. So formation of these somites from this tissue that we call as mesoderm is governed by this network of genes, these 4 genes that we had put here. Wnt is a signaling molecule. FGF is a fibroblast growth factor. It's a signaling molecule secreted outside, bind to protein receptors and regulate gene expression. T and TBX6 are transcription factors, early developmental transcription factors that regulate gene expression. So when mutation in any of these genes or in the pathways governing, driven by the signaling cascade controlled by these factors, yeah, result in complete loss of these repetitive segments from the shoulder girdle below. This is the forelimb. Yeah, from below the forelimb, you see this complete loss of these repetitive units in these mutations. In this, this is TBX6 mutant that is shown here, and the picture is very similar if you look at mutation in Wnt3a, mutation in T, mutation in FGF48, and so on. Okay, so but interestingly, these repetitive units that are formed in the neck region, yeah, these are the embryonic tissue compartments that give rise to the neck muscles. Yeah, they escape the mutation in any of these components in this network. So when you disrupt this network, the neck muscles escape, while the rest of the muscles below the trunk are completely ablated. Okay, telling you that the program by which the neck muscles are formed is different. So these are evolutionarily novel new muscles that are formed, and they seem to have a different developmental program. So this gets, you can say the plot thickens, right? It gets even more interesting now. Yeah. So it doesn't seem to be, it's not like you just took this program and just elaborated this. Yeah, this is one way of doing it. You have a program that is already employed. You just sort of co-opt this program in the new location, and you got yourself new muscles, new set of muscles. But that doesn't seem to happen. Okay, and so this is something that we are probing now. What is interesting that we have a theory here is that, so we are making use of this single cell sequencing data that is already sort of out there in the public domain. Okay, I will not go into the details. So this is basically a type of analysis, bioinformatic analysis, that allows you to cluster cells from the embryo. Yeah, you've isolated single cells, dissociated them, sequenced them. Now, based on their gene expression state, you can cluster them. Yeah, so one cluster means this is one particular embryonic cell type that's going to give rise to one or other adult cell type. And clusters that are closely positioned, neighboring clusters, they share gene expression state. They are similar with respect to their gene expression state compared to clusters that are far apart. Yeah, so these two clusters, for example, are very similar with respect to their gene expression. Yeah, this could mean that they came from the same cell type, that they share a single mother cluster from which they emerged, therefore they look similar, or they develop in the same region. They sort of people who are living in the same household start to behave, in a similar way because they influence each other, or they came from the same parent and therefore they have similar properties. Yeah, that's the kind of argument. So these two clusters that you see are both somite populations. Okay, so at this stage, this is 8.5. So mouse has a gestation of 19 days. This is 8.5 day embryo, where neck somites have formed and a few other somites are beginning to form. Yeah, around the forelimb. So at this point, you see the somites fall into two distinct clusters, which meaning they have two distinct gene expression states. Okay, and one of them, which we think is representing the neck somites, is looking more transcriptionally or gene expression state-wise similar to muscles of the head rather than muscles below the trunk. Okay, so the argument here is like we are looking at a border zone between head and this trunk part. Okay, and this tissue in this border zone, is it behaving like the head or behaving like the trunk? So there is a town close by in Tirupati called the Chittur, which borders Tamil Nadu. Yeah, there are people who speak Tamil. There are people who speak Telugu. Okay, so the question is, where do these people come from? Yeah, whether muscles in the neck. Yeah, so people in Chittur, are they coming from Tamil region? Are they Tamil lineage? Are they coming from Telugu lineage? Yeah, and whoever they may be, whichever lineage they may be, because they are in this border zone, they may have so even if you're from Tamil speaking region, since you're present in the Telugu state, Andhra Pradesh state, you may start to sort of culturally become more Telugu like, right? So this is the interesting thing that happens in the border zone. So very similar in developmental borders as well. So we think these are somites that find themselves in the head-like region, and therefore they have a distinct developmental program compared to other somites. Okay, so it's like a trunk population find itself in the region that is normally, where the head muscles develop, and so it is behaving like now head muscles and taking up the head program. So this is the sort of work that we are currently pursuing. Okay, I think that's where we finish. So this is just to tell you the broad interest as to why this kind of work is interesting. Also from the perspective of human health, there are cardiac defects, facial and neck muscle defects in this group of patients suffering from DiGeorge syndrome. It's caused by a deletion of a small deletion in chromosome 22. They have severe cardiac defects and also these associated neck muscle defects. The connection between these two is now well established because there is one group of embryonic progenitors that give rise to a part of the heart as well as the facial muscles. But why this is also affecting neck muscles has remained elusive. Okay, but we are suggesting that the neck muscle, though it is looking like a trunk muscle in terms of forming somites, because it is behaving like facial or head muscles, it's following a developmental program that is followed by facial muscles, and that may be the reason why this is also affected in this disease. So this is where I would stop, and if there are questions, I would be happy to take that.
Well, it was a.
I have a question.
Yeah, sure.
No, you asked the question. Vidya, you can go ahead. Yeah.
So my question is, it looks like all the processes during embryonic development are very tightly regulated.
Mm-hmm.
So is there any scope for noise in this, or like some kind of, like if there's something that is not orderly, then is there some kind of feedback that can correct this process?
Yeah, that's a very good question. So even if you experimentally, for example, from an 8-cell stage, if you take off one cell, it corrects itself, right? That cell is regenerated. It's not like that embryo will give rise to a baby which lacks certain parts of the body, right? Even at a later stage, slightly later stage, you can remove parts of the body and the embryo can repair itself. So it's called a regulative mode of development. Yeah. So it knows how to correct itself to a great degree. And there is also noise that people, noise at the nanoscale, noise at nanoscale. So transcription of genes happens in bursts. So at any given point, a particular transcription factor can be at a higher or lower level in a cell. So it could be the same cell type. Yeah, you could have come from the same mother. You could be 2 daughter cells, or a bunch of cells came from a single mother cell, right? But within this, you can create heterogeneity simply because of these transcriptional bursts. Yeah. And depending on whether you express this factor at a higher or lower level, a new signal that comes in, you may respond in different directions. So noise at the nanoscale is what actually is one of the mechanisms by which you create diversity. Yeah. So you create heterogeneity within a population of cells by noise at the nanoscale level.
I guess like we don't have any more questions now. Yeah. So it was a fascinating season, and now we are proceeding towards the end of the session. We would like to thank Ram for such an interesting talk. We saw how this body plan is executed, the history of development biology, and how the development biology has helped us in the field of medicine, and how a new emerging field, how we learned about the new emerging field evo. And we also saw like how the neck might have developed during the course of the evolution. So now we are moving towards the end. I would just request everyone who is new to the channel, if you find this video enjoyable and interesting, please go check out our previous videos and subscribe to our channel for more interesting videos and updates. Yeah. So let we just wait for 5 minutes more. Like we should have a screenshot for